interactions with pathogenic microbes. So she completed her PhD from India and moved to Copenhagen University in 2018. She started there as a postdoctoral researcher and is now an assistant professor. Her lab focuses on host-pathogen interactions, disease physiology, varietal resistance, and integrated disease management of cereal diseases, including net blotch and powdery mildew. So today she's going to talk about the combination of non-invasive, high-throughput phenotypings and molecular and cellular physiological approaches to study these regulatory networks on pathogen responses. And so thank you. That's it. And we will be, this is being recorded. So it also asks that you write your questions on the chat. And in the end, we will then start the Q&A session. So thank you all. And Chandana, the floor is yours.
Yeah, thank you so much. So I will start with my introduction. So my name is Chandana Pandey, and as she mentioned, I'm working at Copenhagen University. Recently started as a assistant professor. So in today's talk, I will discuss 3 different points in detail. So the first will be being a plant physiologist. Why do we want to integrate plant physiology at the level of genomics and the phenomics? And that's approach we will take for our case studies, that is the Plumeria and the Perinophora terris. That's a pathogenic fungus and very commonly known for the barley plant infection. So the first topic I will discuss about the integration of physiological phenotyping into the phenomics approach to close the genotype to the phenotype knowledge gap. And then taking this approach, I will discuss the data related to the Plumeria graminis and Perinophora infection in barley using the different barley cultivar. And at the end, I use the biotic stress. In this case, we have used the drought stress, how the drought is affecting the plant pathogen interaction, and how is it harmful for the plant, or is it helping barley to cope up with the pathogen resistance. Yeah, so first I will start with the what is already known in the literature and where we are lacking the information. So there is a lot of information is given how the genotype is interacting with the environment and finally to the management. But there is still a knowledge gap how the physiological response is happening inside the plant or also outside in response to the outside the environment. So that is the major goal from our lab, where we investigate the different physiological responses and trying to connect these physiological responses at the phenotype level or at the genotype level. So as I mentioned, there is a lot of studies had been performed in the level of the genomics, like for example, barley genome. It has been studied very well. There are thousands of breeding lines. There are many phenotyping facilities that can use to assess the plant growth or resistance and all the things. But the point is how we can connect these two parameters from from phenotyping to the genotyping. And this is where the gap comes, genotype to the phenotype. And these knowledge, how we can fill it either by assessing the plant physiology at the internal level or via the external level, how the environment is affecting. And when it comes to the physiological level, what kind of approach we want to take? So we integrate the physiological phenotyping into the functional phenomics to assess the key interface between the genome and quantitative as determinant of yield, quality, or kind of resistance toward the pathogen. And how does it work? For example, the genotype use as a predictor, and finally we use the like for example taking the advantage of precision agriculture at the management later level. And for the predictor of the genotype, that will be the breeding marker, can be related to the different genotype using the metabolome analysis and the different physiological responses. Now comes to the target. What kind of physiological responses we want to see when it comes to the selection? So in our lab, what we focused is the carbohydrate metabolism, and now we are also um, extending it to the other pathway. So the why carbohydrate metabolism? Because we know it plays a very important role from source to sink metabolism, and also when it comes to the pathogen infection, plant feed on the carbohydrate, pathogen feed on the plant carbohydrate. So there is a lot of things which play important role in the carbohydrate metabolism. And finally, after the environmental effect, we also we are very interested to know how the allocation is happening, how much energy is transferring from the plant height or from the source to the sink metabolism, where all the agriculture or farmer are interested, that is the yield production. And when it comes to the selection within the carbohydrate metabolism, so we have published many articles, the lab where I am working, for example, using the extracellular invertase. Here they have mutated the gene, participated in the glucose, fructose breakdown, all the things, and find a very nice correlation in relation to the phytohormone metabolism, cytokinin, gibberellin, and brasinoid. And the second part, how the coordinated regulation of source sink relationship is happening. For example, we have published the data related to the sugar beet plant, where how the carbohydrate metabolism is playing a role to sinking or to go or to take all the things to the sink, that is the sugar beetroot. So this is how we want to take it, this approach. So if we are looking at this figure, so this is how it's predicting. The top label is the genotype, and then the lower bottom is the phenotype. And how do we want to connect this one? And here we want to we usually focus on the enzyme data in relation to the other parameter what we are seeing here. So taking the enzyme from the carbohydrate metabolism, and uh, also with the different antioxidant enzymes. Now when it comes to the enzymes, so we already know there is many spectrophotometer technique where we measure the specific adsorbance of the enzyme activity, but our approach is how we can minimize the sample material, because of all the small level experiment or large level experiment, people are sometimes limited with the plant material, and how we can limit the manpower or to do it in a very efficient manner. So to do this one, first we targeted all the enzymes related to the carbohydrate metabolism. So all the genes what you are seeing in the green color, the enzyme gene encoding the enzyme, are already stabilized in our lab, and that's what we are targeting first at the physiological level, and further if we find something interesting we take it to the functional level by mutating these enzyme coding gene in the plants. So these are the different 13 enzymes related to the carbohydrate metabolism, and these enzymes has been selected from the different pathway, for example glycolysis pathway, for example starch biosynthesis pathway, and also the lipid degradation pathway. So these were the major target we were uh, investigating before. And now comes to the method part. Why this is important, why we are doing it? Because we use the single extraction technique. So we have standardized the protocol where we use 0.5 gram material, 0.5 gram material to 2 gram material, plant leaf or bacterial. So we have standardized this protocol for different plant sample and different kind of materials. So we take the single sample, we do the single extraction, and then we measure all the 13 enzymes from the carbohydrate metabolism. So it's like very efficient technique. And then we use the 96 well plate format. So it's not like single cubette method. We use the 96 well plate from the single extract, and then we have different protocol for each enzyme where we just change the substrate material which is interacting for that particular enzyme, and that is used. And after doing this carbohydrate metabolism, we are interested in many other enzymes. So we also work with all the antioxidant related enzyme. And the interesting fact here, we can use the same extract to measure the carbohydrate related enzymes and also the antioxidant metabolic enzymes. So taking the 100 milligram of material, we can measure 13 C enzyme and 9 different antioxidant enzyme. And we are also working on the different nitrogen metabolic pathway related enzymes. So we have the list of different enzymes which we usually target to connect physiology to the phenotyping. So just so this is unpublished data what we are working, and just to give you the example, and it's a barley hub, so I wanted to show a specific example from the barley. So in this study what we are doing, we have treated the barley plant with different beneficial microbe and growing the same barley at different location in Denmark, which is very different from one location to the different location depending on their soil texture. And then we analyze all these enzymes related to the C enzymes, A enzymes. And in this study, we have also used different kind of barley cultivar. So to give you the one example here, you see, if you see the green color, it's significantly different signature for the enzymes located at different location. And we can clearly see there is a biosignature, for example, PGI, PGM, G6PDH. It is regulated in one barley cultivar from one location to the second location. And then we have connected these enzymes to its functionality depending on their yield or their grain size or their foliar weight, all these kind of analysis. So we do the correlation study. So for example, here the identified enzymes, further we took it to the further next level, and we wanted to see how these enzyme activity in flag leaf are correlated with the enthesis, with the grain number. And we found a very, out of 13 enzymes, 2 enzyme which is very nicely related to the grain number. So it's again, it's not getting, so we find a very specific uh, I would say the biosignature related to the enzyme. And then we target those particular enzymes for the further study or studying the other kind of work using that particular knowledge from the previous book. And this is again the second example where we have identified the biosignature, where barley is growing in different soil types, for example, loamy soil, sandy soil, clay soil. So we have the barley cultivar growing at different location. But what are the target in the plant? How the physiology in the plant is working? Then we have identified the particular enzyme which is responsive in that particular soil type. So that's where we want to connect with the genotype, that is the barley cultivar, and also at the later stage of the phenotype, that can be the yield or visible appearance or the height, all kind of phenotype thing. And then we have published different article where we have shown how the high throughput phenotyping is correlated with the physiological response. Elevated carbon dioxide is related to the physiological response related to the carbohydrate and antioxidant metabolism, how the barley resistance against the pyrenophora is related to the physiological response, which I will discuss more in detail later, and further the yield and also the pathogenic strains. So how all these are correlated with the physiological response and what are the particular enzymes or signature are related. And advantage of having these one, it's like very fast growing, and we can do a lot, like more than 10,000 enzymes within a week, 10,000 samples within a week. So that is very good advantage of using these techniques. Now, after discussing this whole physiological thing, we have this setup where we measure the carbohydrate, antioxidant metabolism at the physiological level. We also test the plant at the phenotyping facility. So nowadays, it's very famous, sensor-based phenotyping, all the things. So we also have this facility that is called sensor-based automatic phenotyping here in our campus, Copenhagen University. And this is the conveyor band based automatic high throughput phenotyping under the control greenhouse condition. And what you are seeing here is a real picture from the campus, and these are the different fixture you see, and this is the graphical representation. Here we can stand and then we see all the plant fixture within this facility. And here we have the automatic water supply and the water station. If we want to irrigate the plant, it can be irrigated as many times we set up in the system. We just tell the machine, okay, we want this watering supply, and then it take the system and it goes automatically. And how do we do the phenotyping facilities? So for the phenotyping facility within this vision station, we have the LED lamp which measure the different kind of parameter, which I will discuss in detail. And in total, we have 117 big pot space, but one pot can be divided into the 4 small pots for the Arabidopsis plant as well. So it has automatic. We can also randomize all pot together. So it's not like plant is standing at one position. It can also randomize during the period of day. Okay, so within this phenotyping facility, what do we measure? So we measure these LED light can measure the different reflectance wavelengths, and that is the goal here, to connect the specific signature for these reflectance wavelength to the physiological parameter related to the enzymes or to the molecular parameter. So these are the 10 different narrow banded LED which we measure for each plant, and we also have different fluorescent filter. For the moment, for my study, I'm using 3 different emission filter together with the UVA and blue excitation range. And once we get the data, we analyze it via the Videometer software. And when it comes to the pathogen response, so here you see the light, blinking light. It's a camera where the camera is located. So this is the normal plant which is standing inside the vision station. But when it comes to the infection, so we also want plant leaf to be visible or to get the proper imaging. So for my study, for the barley study which I'm presenting today, we have mounted the leaf on this plexiglass, so the camera will get the flat surface, and it can analyze the data, or it can detect the very fine reflectance or fluorescent matter with the very small sensitivity. Yeah, so if we are investing now, after this, the second question comes: if we are doing or spending a lot of money to build up a big phenolab facility, so why not to do this thing at the microscopy level if the university has the microscopy? So that's why we were also interested to compare the data from phenolab to the microscopy? Can we see the same kind of result, or can we mimic the result, or how efficient is to work with the microscopy targeting particular reflectance or the fluorescent wavelength? So for this study, I targeted these different reflectance wavelength and also the fluorescent, that is the emission spectra, 520, 690 and 740, exactly the same what we have in the phenolab. So now comes to the result part. So now I will discuss about the differentiation of Plumeria graminis and the Pyrenophora terris barley resistance. So the thing what I discussed, so these are the approaches we use to study most of the work in our lab, using the physiological responses, phenotyping responses, and finally the genotype responses. So first comes to the introduction, what pathogen are in which I am working. So these are the two different pathogens which are very responsive to the barley cultivar. First one is the barley biotroph, and the second one is the hemibiotroph. Biotroph is Plumeria graminis, very well known, and it causes the powdery mildew disease. And the second one is the netch blotch disease, a netch blotch disease caused by the Pyrenophora terris. So for the Pyrenophora terris, we have already published this data, and in this work we have used 4 different barley cultivar, and these barley cultivar has been tested against the Pyrenophora terris for the reflectance, fluorescence, and also the physiological parameter, carbohydrate metabolism, antioxidant metabolism. All the enzyme which were very interesting, we tested it for the gene expression analysis. And further, in this study, we have also tested the temporal dynamics, how the physiological responses are changing over the period of time, starting from 8 hours after inoculation of pathogen to 120 hours after the inoculation. And it's very interesting to see how the responses, enzyme activity is changing from very early time to the late time point for the resistant to the susceptible cultivar. And finally, connecting these to the reflectance or fluorescent, that is the phenotyping. And this is just one example. So this guld is the barley cultivar, and it is very resistant cultivar for the Pyrenophora terris. And here we observe the very early activation of defense mechanism by increase in the activity of all the carbohydrate metabolism and the antioxidant metabolism, which is shown here. And further we tested the different gene which were involved in the defense mechanism, and we found the same kind of pattern. So it is also very interesting to see how the robustness of the enzyme activity is working, because the gene, we know the gene changes its response very fast, but still we see the correlation from gene to the enzyme. So that was also very interesting finding. And now comes to the phenolab data. So as I discussed, we have 9 different wavelength. You see here 365 to 970 nanometer. So an MS blood plate, it's a susceptible cultivar and gold. And when we observe all the data and analyze the data, so yeah, it's a complicated figure. I will discuss more in detail for a single wavelength. But here you see the green color is appearing very early, means there is a very higher value for the reflectance value in the susceptible cultivar as compared to the resistant cultivar. So again, we find the specific wavelength which were specific for the symptom for Pyrenophora terris, only present in susceptible cultivar, but it was not present in the resistant cultivar. So in future, if we want to utilize the particular wavelength, so we can select, okay, this particular wavelength, we can go further for the pathogen study. So after studying this one, we found out, okay, resistant cultivar, we identified the gold, susceptible cultivar, MS flat plate. So we were interested to see what is happening if we use the most resistant and the sensitive cultivar for the Plumeria graminis. So we took two most interesting cultivar from the Pirinophora terris study, and now we tested it for the Plumeria graminis infection. And to plan this experiment, we have divided our experiment in two different parts. First, we wanted to see what is the comparison for the macro and microscopy sensor based phenotyping, that is comparing the microscopy to the macroscopy. And the second part is, because we already know, okay, drought pathogen has impact on the resistivity and sensitivity of cultivar, but how the drought is affecting the cultivar. So that was the major question in the second part of the study. And to give the drought treatment, we started the drought as a pre-drought treatment. So we started the drought at 12 days, and at 14 days, after 2 days of drought treatment, the Plumeria infection was uh, performed. And again, this experiment was performed in the phenolab. This is how the leaf were mounted on the plexiglass. And after this, we harvested the sample at 8 hour, 24 hour, 48 hour, 120 and further. And for the phenolab analysis, we have analyzed the data each day 2 times. So reflectance and the fluorescent value we have for 8 hour, 24 hours. So that was very nice to have the automatic system. Now comes to the result part, and here what we see, that the symptom development allowed to discriminate the susceptible gold and from resistant MS cultivar for the Plumeria graminis. So if you will observe here what I mentioned before, that for the Pirinophora terris, gold was very resistant, and but for the MS blood, for the Plumeria graminis, gold is very sensitive. So that's how we got attracted to this project. Okay, one is hemibiotroph fungus Pirinophora terris, which is infecting gold, and there is no symptom. But when we infected this gold with the Plumeria graminis, that is biotrophic fungus, we see full of symptoms. So means how these two pathogens are interacting with the plant, or what are the functionality, or what are the target in the plant which making it susceptible for one and the resistant for the one. So when we analyze the phenolab data, so this is pseudo RGB data. So we checked these data and quantified, and what we found in the initial result, that when the drought was given to the very sensitive cultivar, so for the Plumeria graminis, gold is very sensitive. Within 3 days, we see full of symptoms. There was a less symptom in the gold cultivar as compared to the MS blood plate. So gold is very sensitive, MS is very tolerant. But what drought is doing, drought is making gold to the less sensitive, and the resistant cultivar to the more to the less resistance. So that was very interesting, and that's where we wanted to investigate why this is happening, why the sensitive cultivar is becoming less sensitive and the resistant is becoming, how the drought is interacting with the pathogen interaction. And now it comes to the phenotyping. So the result is divided into three different parts. First, we started with the phenotyping, then to the physiological responses, then to the molecular responses. And within phenotyping, we are comparing the microscopy with the phenolab. When we perform the microscopy for the specific wavelength, that is reflectance wavelength, 525, 570, 645, and 670, what we observed, that there was no early signature for the disease resistance in these cultivar, especially for the sensitive cultivar. Here, we only observe changes at 5 day and 6 day, where we see that the sensitive cultivar has higher reflectance as compared to the mock or the resistant cultivar. So these four lines, resistant and sensitive cultivar for their mock and the infected plant. But Out of all these, we observe that the fluorescence imaging for 520 nanometer, here we were able to detect the symptom even when it was not visible via the eye. So even after 1 day after infection, we found the signature that was the 520 nanometer emission wavelength. In the blue excitation, there is a increase in the reflectance wavelength starting from a very early stage. So that was the interesting. But the only thing here, we just got one biosignature for the pre-symptomatic detection. But when it comes to the work, microscopy is a lot of work. We have to handle each sample, make each slide, and do a lot of work. And we found not a lot of biosignature for the early detection. But the interesting thing, what we found here, that we measure the reflectance in the area where we can't see the visible symptom. So for example, the leaf infected with the plumeria, uh, we took the whole leaf and then selected those areas where we can't see any symptom, and we compare the both uh, resistant and the sensitive cultivar. And here what we found interesting, that in resistant cultivar, that one is the MS black plate, we see higher reflectance at 3 days after inoculation. So that's what we are studying in detail, means higher reflectance 3 days after inoculation in the area where there is no symptom can be the reason that here some defense mechanism is happening, or that's where the resistant cultivar is activating its defense mechanism at 3 days after inoculation. So that's what we are investigating more in detail using the physiological phenotyping. Now comes to the phenolab. So after investigating the microscopy, we found, okay, we can only detect at the later time point with most of the reflectance and the fluorescence filter except one. So we wanted to see what is happening in the phenolab. But with the phenolab, interesting thing here was that we were able to detect identified 7 out of 9 reflectance wavelengths as a marker for the sensitivity of the Plumeria graminis. So in the bull cultivar, this is very sensitive cultivar, and you see this red color with the higher activity or higher reflectance value, which is absent in the MS flat plate, and that was activated very fast starting from 2 days after infection. So that was a great success to identify the early detection. And further, so the previous slide here, what you are seeing, it's for 9 different reflectance wavelength, different time point, and different cultivar. So it's a little bit complicated. So that's why I made one single graph to show you how does it look when we select the single wavelength. So for example, this is 25 nanometer, and you see this gold cultivar, it is heavily infected with the Plumeria graminis. That is very rare to see with the eye at the initial phase after 1 day after infection. But when we use the phenolab system, we were able to identify the particular marker, that is 525 nanometer, uh, to see the increase in the reflectance wavelength. So we can use this particular wavelength for the further investigation. And these are the wavelength which show the same kind of pattern what we are seeing with the 525 nanometer. But there was no impact of 780, 890 and 970 nanometer for the sensitivity of cultivar. Now comes to the fluorescence signal. So here we were able to identify the increase in the fluorescence signal in the bull cultivar and also in MS cultivar. So at the later stage, we also observed that the resistant cultivar somehow get some kind of a little bit of infection, but not, I would say there is no filament formation. So we see it after 5 days after inoculation. So in phenolab, we can clearly say that reflectance is working well ahead of fluorescence signal. We can do the pre-symptomatic detection. We identified the different signature related to the reflectance wavelength, and for the fluorescent signal we identified it only after 4 days after inoculation of pathogen. Yeah, so now if we have to compare the phenolab with the microscopy, what we see the same kind of pattern, that the sensitive cultivar has higher reflectance or higher fluorescent value as compared to the mock or as compared to the resistant cultivar. But the difference here is phenolab is very high throughput phenotyping. Here we don't have to be in the lab, we insert the plant in the chamber and then it goes for the automatic. But yes, when it comes to the data, we need to handle a lot of data and do the stats analysis for the big set of data. And for the microscopy, it's time taking. And again here with the microscopy, if we are talking about the Plumeria graminis infection, so infection is like a powder formation. So while we are targeting the microscope, we have to be very careful for the selection of the region. What region we select or put inside the microscopy lens. But with the phenolab, we measure 10 leaf at a time, and it is also very clear with the picture. So here I have added one picture. In the green leaf, you with the eye we can't see any visible symptom, but when we perform the selected reflectance as a biosignature and we did the marking, so we clearly see the different spores laying on the leaf. So that is very interesting to see at a very early stage. Now it comes to the how drought is affecting the sensitivity of cultivar. As I mentioned before, we found that gold is very sensitive for the Plumeria graminis. But when we provided the drought stress, the symptom was less or the sensitivity decreased. But in resistant cultivar, where we cannot see any symptom even after 5 days after inoculation, we see there is a increase in the sensitivity. Means we see more symptom when the drought was there. So there is two things. One, why drought is affecting one cultivar and making it less um, less healthier, and why it is making more healthier to the other plant. So that's what we were interested to see, and can we see it with the reflectance and the fluorescent data as well. And we were able to see, yes, out of 7 wavelength where we identified, okay, these wavelength we can use for the pre-symptomatic detection of the pathogen selected, and we found few out of 7, 3 were able to detect the drought with pathogen as compared to the pathogen only. For example, 670 nanometer, here at the later stage we found there was a decrease in the reflectance. If we are talking about the decrease in the reflectance, means less symptom. So more symptom will have the higher reflectance. If it's mock plant or the tolerant plant, then it will have the less reflectance value. So we found, okay, in the gold sensitive cultivar, reflectance was decreased, but in resistant cultivar, reflectance was increased. So again, it's proving our hypothesis, and that's where we wanted to connect our physiological data. And these are the some detail where we found, okay, these are the. Now comes to the physiological phenotyping. So we identified, okay, gold is sensitive, MS blood plate is resistant, gold is becoming less sensitive when drought is there, MS blood plate is becoming less resistant when drought is there. So we wanted to see what is happening with the antioxidant enzyme. And what we found out that in resistant cultivar, uh, most of the most of the enzymes were activated very early, 8 hours after infection. That was missing in the susceptible cultivar. So means there was an early activation of defense response in MS blood plate, which it fight against the pathogen very initially, uh, and it maintained to the later time point. But in gold cultivar, we found it was not activated early, but it was in the later time point, which further disappear at the later time point. So that's the reason it is making it susceptible, and now we are working to find out the specific target where it is interacting with the pathogen. Now comes to the Plumeria plus drought pathogen, and what we observe here interesting in the gold cultivar, where it's making susceptible to the less susceptible, almost all the enzyme activity were upregulated when the drought was there. So again, it's proving our hypothesis, what we observed previously, that DHAR, APX, it can increase the enzyme activity of these enzymes when there is activation of defense mechanism. Yeah. So this is one example where we see in MS blood plate, GR activity has significant increase in the Plumeria graminis treated plant as compared to the mock, and they are significantly different from each other, significantly different. So this is just one example that we have tested it at the stats level as well. Yeah, and these are the different enzymes we have investigated and found as a target from the antioxidant and the carbohydrate metabolism. So I'm not showing the other data related to the antioxidant, but I just listed few enzymes which were popping out when drought is there, means when we combining two different stress. Further, we were interested how the phytohormones. So we know all these enzymes has also connection or regulated by the phytohormone pathway. So we just to find the very preliminary data, we also investigated the role of phytohormone, and what we found out that the gold, it's very sensitive for the Plumeria graminis. When it was combined together with the drought, the jasmonic acid was increased, again making it making the defense activation by increasing. So drought was able to increase the jasmonic acid content in the infected leaf. And when it comes to the abscisic acid, what we observe in both gold and MS, ABA was less in gold and higher in resistant cultivar. But when we combine it together, what we found that gold has higher abscisic acid as compared to the MS blood plate. So it turns when it combines together that gold has higher, MS has lower, while when the individual stress was there, so it was other way around. And now we have also connected the transcriptome data to validate these pathway with the ABA, what I mentioned before, with the jasmonic acid, and also the interesting enzyme what we detected via the carbohydrate and the antioxidant metabolism. And we have investigated, we have performed the transcriptome analysis of Plumeria infected plant, Plumeria plus drought infected plant, and also the Pyrenophora terris. Because here again, we also want to compare these two pathogens, how it is affecting the same cultivar in two different manner, what are the functionality or what is happening inside the plant to make it tolerant or sensitive. So we are investigating that, and this is the pathway what we have. We are checking it and we are proving it using different method, using the transgenic and all these lines. So again, this is unpublished data where we are working right now to prove the hypothesis how the enzyme working or downstream, it's regulating the downstream pathway in the defense mechanism when it comes to the Plumeria infection or the resistivity of cultivar, or when it comes to the combined effect. So for the combined effect, yes, ABA is very important for the drought, and that's where we are also finding it that Plumeria under drought stress pathway is regulated by the ABA pathway by a different downstream pathway. Yeah. So after finding this one, okay, with the phenolabs, we can identify the different resistant cultivar, different sensitive cultivar. We can identify the impact of drought on the phenolab, physiological responses. Then we have also checked in our lab how the different resistant cultivar is behaving. Can we use the phenolab to identify the signature, or can we find any differences in the resistant MLO or MLA resistant based phenotyping? And it is already published in Plant Science, where we have identified the automatic classification of the powdery mildew resistance mechanism for MLO, MLA by the support of vector machine classification with high accuracy. So we use the phenolab data, and then we do the different machine learning, automatic classification. So for example, right now we are working with different barley breeding lines to see how the beneficial microbe is changing the phenotype. So, and we are able to find the particular signature from the carbohydrate metabolism, and we are also testing it into the phenolab to connect particular wavelength to the physiological parameter. So now when it comes to the summary, so from the barley and Pyrenophora terris, what we observe that the sensor-based phenotyping allow to discriminate the sensitivity and the resistance responses both by phenolab and the microscopy. Second, drought stress is differentially affecting the sensitivity or resistant cultivar, making resistant less resistant and sensitive to the less sensitive. Third one is the biosignature for the sensitivity of registered resistance is same for both hemitroph and the biotroph. Means when we find the same higher reflectance in both Pyrenophora terris and the Plumeria graminis sensitive cultivar. And further, we identified the enzyme fingerprint related very specific for the Plumeria graminis and also very specific for the Pyrenophora terris. And when we combine Plumeria graminis with the drought, so we have the other. And further, we identified the functionality, how these interaction is happening at the functional level, where we are identifying, we are working more in that direction to find the upstream and downstream regulator in this pathway. So at the end, I can conclude, okay, yes, by using the phenotyping, physiological phenotyping, where we use the enzyme and also the breeding lines or the genotyping, we can fill the gap uh, and identified many more signature which is still missing in the community. Yeah, and finally, we can fill the gap from genotype to environment and the management by the integration of physiological phenotyping into the non-invasive phenotyping to assess the phenotyping plasticity in the multifactorial environment, for example, beneficial microbe, pathogen, drought, heat, all these things. So what we should remember after this presentation, yes, the plant biotic interaction in relation to the primary carbohydrate, antioxidant metabolism, beneficial microbe, pathogenic microbe, and the hormone play an important role and can be considered to connect the phenotyping, genotyping, and work as a bridging gap here. And we also have analyzed the metabolic data to connect the carbohydrate from source to sink relation. And finally, linking all these data together to make it sense. Yes, how it's connected from very early top level to the bottom level. Yes, so that is the end of my presentation, and I would like to thank all my lab members and also the people who initiated the work, for example, Dominic, and who helped me throughout the analysis, for example, people who helped me for the transcriptome analysis. And at the end, Thomas Roich, the lab where I'm working, is a physiologist, and I'm working with him in his lab. So a great thank to him, and Alexander Schulz, who is helping me with the microscopy, and we are also writing the manuscript where we are comparing the phenolab with the microscopy. Yeah, thank you so much.
Thank you so much, Chandana. Very comprehensive work. So I open the floor to questions. Can you please either put it in the chat? I haven't seen anything yet in the chat. Okay, yeah. Robbie, do you want to say it, Robbie, or do I have to read it? I have to read it. Thank you. Okay, Robbie, thank you very much. Nice talk. Perhaps I missed, but wasn't clear for me was how you propose to use the data that you generate as biosignatures. And secondly, do you think it is possible to draw general conclusions from comparing only two genotypes?
Yes. Yes, so how we identified the biosignature, because when we compare the resistant to the sensitive cultivar, or if I'm talking about one pathogen to the second pathogen, we have very specific enzyme activity, which is only regulated in the Brumeria graminis, but not in Perinophora terris, but not in other pathogen. So it is showing very specific activation only in presence of that pathogen, not in other. And how does it different from one cultivar to the second cultivar? Yes, if we see the activation in resistant cultivar, it is not activating in sensitive cultivar. And the interesting thing here, we have the same cultivar, the gold, which is very sensitive for the Plumeria graminis, but it is not sensitive for the Perinophora terris. And we see same, for example, we have the PGI, it's a polyethylene glycoisomerase, which is only regulated in sensitive cultivar, but not in the resistant cultivar. So I would say it is also a very specific for the cultivar.
So I'm going to ask the question out loud
Yeah.
now. So did you look at many resistant cultivars and you always get the same response?
So initially we started with the 9 cultivar. So out of 9 cultivar, we found 4 which was showing the symptom and not showing the symptom. So which were growing properly. So for the moment, specifically for the pathogen, we selected 4 cultivars where we found 1 resistant and the 3 sensitive cultivars. So this is the maximum number we have tested.
Yeah, well, I'm trying to work out in my own mind. Sorry if you've explained it and I'm not getting it, but if you take 4 resistant cultivars against Plumeria,
yeah,
and you generate your biosignatures or your physiological phenotypes, do all of the resistant or all of the susceptible cultivars show the same biosignature or the same physiological phenotype?
I would say not all will show, because we have other study not related to the pathogen, but we have different breeding line from the barley, and we are using the single beneficial microbe which is changing the root architecture. And what we are finding out, that the group of breeding line which showing the change in the root architecture shows the few enzyme which is always popping up or which is always showing the response, but some of them are very specific for those genotype or the breeding lines. So I would say it depends, are we treating it just under the mock condition, and or how much influence it has from the external environment. If it's pathogen, so I would say there will be the influence of pathogen on the this biosignature. But if it's resistant cultivar, so we will see the biosignature for the resistant cultivar, and which will vary from one to the other, because when we work with the beneficial microbe, we don't see any changes in the phenotyping for few cultivar or for few barley genotype, but we still see some signature, and that's where we are interesting to know how it's communicating with the plant, how bacteria is communicating with the plant, what are the responsiveness in the genotype or the different barley genotype, or how it evolved from very historical old line to the develop over the period of time. So that's what we are also approaching in that direction.
Can I continue to this discussion, or just a very simple question?
Mm-hmm.
If I gave you a barley genotype that you didn't know was resistant or susceptible,
Yes.
and you established its bio
Signature.
signature, could you tell me if it was resistant or susceptible?
Yes, that's what we
Okay.
have observed
Okay.
till now. Yes.
Thank
And,
you.
and the good thing here, it's not very complicated method, and it's very robust method, and we see it even from one time point to the second time point and over the period of time. And that's why I showed one example that we found the same kind of biosignature varied from different land in Denmark. So we found the biosignature for the beneficial microbe in barley cultivar located at different location. Means all the community in the soil is not changing the biosignature. It's only one single microbe which is regulating that, or I would say the responsiveness of that particular cultivar. Yeah.
Thank you. Thank you.
Cool. Any other questions from the audience? Well, maybe I can just ask one question.
Yeah.
So I was very interested on your plexiglass
Yes.
that you basically flat and then
Yes.
you screen the plants. Now, does it break the leaves, or how are the leaves not detaching and breaking? Because we handle also barley plants, and sometimes just the shaking of the conveyor belt can just make sometimes the leaves detached and all of that. So I was just curious to see how do you think your plexiglass system is working?
So I would say if I'm dealing it for the first time, or if
Okay.
sometimes it's very complicated method to do, so we took help from the student as well to do the mounting same day. So what we do the mount, and we use very soft plastic, the rubber band, the black color. And in my experiment, I managed to exclude all the plant where I saw the mechanical injury. But and that's why we also put do this mounting, what we say mounting of leaf on the plexiglass 2 days before, to make the plant that it is stable. So the day I
Okay.
started the drought, we do 2 days before that plant get
Okay.
healthier. But there is a way to handle this, and I would
Okay.
say no
So
matter.
you basically, if by any chance a leaf is broke down, then you
Yes.
don't analyze it because you did 2 days before. Okay.
Yes, yes.
Would
And
also the plexiglass affect the transpiration of those leaves? Do you think that would impact overall the development of the symptoms, because you're basically blocking the stomata on
yes.
those leaves for a couple of days?
Yes, so I would say yes, 100%, but we also do it for the mock plant. So it's not only the infected plant, because we are comparing the same kind of mechanical injury, same kind of situation for the plant. So that's why, yes. So to answer your question, yes, but the data what I'm showing, we have used the same setup for the mock or resistant sensitive cultivar.
Okay. So I think that I saw a hand up from Salar. Was it?
Yes.
Okay. Hi, Salar.
Thank you, Sonia. Hi, Sonia. Thank
Nice.
you.
Hi.
Thank you, Chandana, for your nice talking.
Mm.
Actually, I have a question
Yeah.
about the part 2 of your
Mm-hmm.
presentation regarding the drought stress.
Yes.
You showed a slide, I think, where you indicated the time point where you applied stress,
Yes.
drought stress.
Mm-hmm.
I don't remember which time point was, in which growth stage
So
you.
yes, for the drought stress, when we planned the experiment, we thought it's good to, if we want to combine the stress, pathogen and drought, so it's good to have drought before, like predisposition of the drought. So it was, so drought was given at 12 day of the plant growth. So plant were started 0 day, the germination, and then it was grown until 12 days. The drought was given and it was continued till 2 days to get the certain drought regime. And after 2 days, we infected the plant. So it's pre-drought treatment after 12 days.
Okay, so my main question is this.
Mhm.
When we would like to evaluate many cultivars
Yes.
for this kind of experiments,
Mhm.
of course, maybe they are different in phenological
Mhm.
stages. For example, the flowering or the plant height
Yeah.
is phenological. So how can we reveal the responses of cultivars independent of uh, escape mechanism, which may be caused by phenological uh, stage of the plant?
Yeah. So, but
Because
you are talking about specifically for drought stress
maybe,
or
maybe,
just the
maybe, maybe,
biosignature?
maybe, maybe a genotype that is
Mhm.
seem to be drought tolerant. It may be due to its escape mechanism. So how can we distinguish these two uh, responses?
Yeah, so actually what we do, just to give you one example, so we are also working with the maize plant and we took the cultivar from the Spain. So we tested the field study in the Spain and Denmark as well to find out the biosignature under the drought stress and also under the normal condition. So the Spanish cultivar, when we took it to the pheno lab, we grew it together with the Danish cultivar. So we found out that the Spanish cultivar was not growing properly or the germination was late because we also mimic the soil condition here in Denmark. So
Yeah,
yes, we skipped the uh, 14-day harvesting because our priority was to find the biosignature for the drought resistance, not the growth parameter. And we were able to find the same biosignature here in Phenolab using the soil from Spain and also in the field. So that was very interesting. So even
yeah,
it is growing very fast in Spain, growing slow in normal greenhouse condition, but we found the same kind of signature. So I would say it depends on the question what we want to ask, what are we targeting in that sense. But yes, that will affect the result, I would say. If we are not going, for example, if I want to do the pathogen stress, so I usually mount the second leaf of the plant. If I will not get the second leaf, so I can't compare those two cultivar. So I have to wait
yeah.
or get the situation where it can grow faster. Yeah,
Okay. Thank you, Chandana.
welcome.
Thank you. Any other further questions? Because we are, think, yeah. I'll just thank you so much, Chandana, for giving such a
Thank
nice
you.
talk.
Thank you for giving me the
And
opportunity.
good luck with all of your
Yes.
experiments that you have ahead.
Yeah, and just to say word here, so we are doing a lot of biosignature, dealing with a lot of breeding line, and I know this is nice place to advertise. So if somebody is interested to know or to work or collaborate for this high throughput or semi high throughput enzyme activity, so we are very much interested. Yeah.
Well, that's very good. Thank you so much.
Yeah.
Thank you.
Thank you.
And thank you all for joining, and hope to see you soon in 2 weeks' time, right, Robbie? I think it's 2 weeks' time. 4 weeks' time. Oh, because of the summer break. Yeah.
Yeah.
Wait a minute. I think I have something here from Joanne. Joanne, you have a hand up? Or were you just waving goodbye?
No, I was just waving goodbye.
Sorry about that. I
The
thought.
wrong hand.
Okay, thanks, Joanne.
Thank you.
Bye,
Bye.
y'all.
Yeah.
Bye, bye, bye.
Bye
Thank you.
bye.
Bye. Bye, Chandana. Thank
Bye.
you. Bye, bye.